I have lured you all. Lured you into my Sonic Hedgehog party. The fancy video game name, the fancy hedgehog name has roped you into my extremely weird and extremely complicated lecture about this, like, really vitally important protein that just, it's called Sonic Hedgehog. That's its name. There's nothing I can do about it, and there's nothing anyone can do about it. Oh my god. Hello, Gray. I'm just so ecstatic to have lured you all here with this shiny video game character. You see, when it comes to molecular, developmental biology, proteomics, genetics, anything, if you discover something, you get to name it. You pretty much get to name it whatever you want. You can name it Sonic Hedgehog, as you can see. So it's going to be super, super fun. In my explanation of what this protein does, why it's so important, and why it's freaking called this, we're going to secretly talk about a lot of other super secret concepts that are deeply entwined in developmental biology, which is 1/3 of my bachelor's degree. It is going to be a grand old time. So let's start with, why is it called this? Why do we call it this? Why is this godforsaken video game character that makes us a nervous system called Sonic Hedgehog? Like most genes, especially genes that are involved in development, it was not discovered first in humans. It was discovered in fruit flies. Drosophila melanogaster is where Sonic Hedgehog was first observed by a woman named Christiane Nusslein-Volhard. She is the only person from Germany who has won a Nobel Prize in science. The only woman from Germany who's won a Nobel Prize in science. So she discovered this gene in flies. And oftentimes when we discover things like that in other model organisms, like sea urchins, frogs, and such, anything like that, the next thought is like, all right, where's the human gene? What's the homologue in humans? The gene that does whatever it does in flies, there's got to be one that does it in humans. So there was a group of people looking for one in America as well. And they were still looking in flies, though. That's always where you start. And often you name things after what, when you name a gene, it's after, like, the disease that it causes. So, like, all of us have the POF gene, primary ovarian failure. Not all of us even have ovaries, but we all have the gene because the mutation in it causes ovarian failure. With these guys, Sonic Hedgehog mutations cause freaking spikes to be all over them. Just like a little hedgehog. The little flies got turned into hedgehog because they were made incorrectly because of this mutation. So despite the fact that humans and flies are completely different, we actually have 60% homologous genes between each other. That's how, like, a lot of genes that are for making the body like Sonic Hedgehog, they're kind of ubiquitous throughout all life, which is really fascinating. Level 2 mutant fly, exactly. You get mutations in Sonic Hedgehog, you could pop your spikes. It's lit. I wish that this is what it did in humans, but unfortunately it does really effed up. Mutations do way more effed up stuff in humans. So this lab in America did discover Sonic Hedgehog in humans, and we discovered 2 other hedgehog genes too. In addition to Sonic Hedgehog, there's Desert Hedgehog and Indian Hedgehog. All 3 of them are important for human development. It's just Sonic Hedgehog is, I mean, it's the flashiest, and it is objectively, like, the most important. I'm just gonna read you an excerpt, honestly, from this is an interview with the head of the lab that discovered Sonic Hedgehog in humans, Cliff Tabin. The hedgehogs, the genes, were initially named for the bristle appearance of the Drosophila larvae carrying mutations in the fly hedgehog gene. Once vertebrate hedgehogs were discovered, Cliff's group provisionally named them after a different species of hedgehogs. A different species of hedgehog. Sonic Hedgehog, in fact, was originally named Common European Hedgehog. Boring. Bob Riddle, the postdoc in the Tabin lab responsible for initially cloning the gene, requested the name be switched to the much livelier Sonic Hedgehog, after the Sega Sonic the Hedgehog character from a British comic book his 6-year-old daughter owned. The postdoc thought the name was appropriate since the Tabin lab had a reputation for being loud and boisterous. And the postdoc played guitar in a band. I don't know why they included that. Cliff's collaborators hated the new name, however, since Sonic meant groovy when they were growing up in Liverpool. The name was changed anyways, and in a funny coincidence, Tabin's paper on Sonic Hedgehog came out the same time Sega first started promoting its Sonic the Hedgehog character game in the United States. A naive colleague, after seeing a McDonald's promotion for Sonic the Hedgehog, called Cliff, shocked that the Tabin's Lab new discovery was so popular that McDonald's was basing Happy Meals off of it. Yes. It was so fortuitous. Like 1995 or whenever this was a crazy time for things like this. I mean, it's nutso, and I love it. So hilarious. The main takeaway from this is that it's hard to study development in humans. And it's a lot harder to get permission to do like petri dish fertilizations of human embryos than it is for frogs, sea urchins, zebrafish, and other organisms that are not human. It's actually funny that you bring up copyright issues with gene names because we're going to talk about a little something something later. It is kind of funny that like Sega doesn't mind, because technically they have the rights to it, but they probably think it's cool. But it is easier to study these things in flies versus humans because you can have like a bajillion flies and their body plan is a lot simpler, so it's a lot easier to study. But the fact that these mutations give them spikes sort of hints at their importance in making our body plan. So it's expressed in a lot of places during development, but its main thing is it establishes our ventral and dorsal axes, front and back. And it also establishes a left and right axes as well. So that's important. Knowing like where everything is supposed to go on an axis when it comes to development is you want to get that correct. And it also in those areas determines cell fate, cell proliferation, cell migration, anything else that's important for getting cells and therefore eventually tissues and organs where they need to be in their final positions by the time birth happens. So firstly though, I'm going to be discussing mainly Sonic Hedgehog and the central nervous system because it does do certain developmental things in other areas, but its role in developing our brain and spinal cord are the most studied. So that's what I'm going to be going over. And before we do that, we're going to go into some developmental concepts real quick. So let's take a look at this egg I just fertilized. Day 1. Fertilization literally just happened. All the genetic material is in one cell. And it's in one diploid cell. So all the genetic material is together. We're going to skip ahead to day 19, though. Day 19. What's special about day 19? Day 19 is when gastrulation happens. Gastrulation is the development of the 3 germ layers within our developing embryo. And those layers are important because they're going to become their own specific populations of specialized cells that are going to become, things that we're going to need when we're born. It's a huge milestone in embryonic development. And when we're at day 19, this is going to be a couple days after the fertilized egg, the zygote, has started like embedding itself in the uterine lining. And the cells that are going to become the baby and the cells that are going to become the extra stuff, like the placenta and shit, are separating from each other. And as the germ layers are beginning to form, I'll also point out, where's my pen? What we're looking at over here, obviously this is a very, very, very early embryo. These, Jesus. Do I have to tell you again to be my pen? These cells over here are going to be what's called like the extra embryonic stuff. So the placenta, the amnion, things that are going to support the fetus, and sort of, not like literally, but kind of like connect it to the whoever's carrying the baby. This area is the embryonic tissue, and the part that we care about right now is this little stripe right here. This is where cells are gastrulating, basically. And what's important is this area. Oh my gosh. About this area right here. It's really hard to write with this pen, I guess. Is where we have the primitive groove. So we have that over here. This picture is really, really, really amazing. We do love human embryos. They're like so adorable, little like flat, little ugly, little attachy, crazy things. But the primitive streak drawing right here is really good because it's showing you that these cells are actually migrating inwards. And it's a little hard to imagine because embryos, yes, flat and parasitic, literally. Because there were other like really cool slices, but I hope you guys can see my cursor good. But like these are like little tendrils, like grasping into the uterus. And the zygote itself, it doesn't just attach on the outside. First, it goes completely inside the uterine wall. It's freaky. But during this stage, these cells are migrating sort of inward as the front and back axis of the body is being created, and that's where we're getting our 3 different germ layers, and they're very important. I'll tell you what each of them is going to become. So we've been here. The first one is the endoderm. Endo meaning inside, so it is the most inside layer in the developing embryo at this point. It is going to become the linings of a lot of our tracts, our respiratory tracts, our GI tract, but not our mouth or our ass. That's a different thing. It's going to become the lining of our ear canals, and it's going to become the lining of the inside of our, like the straight up inside of our lungs as well, and our bladders. So, I mean, the key commonality there is it's body linings, inner linings. The next germ layer is going to be the mesoderm. Meso meaning middle. So it's going to become the outer layers of things like the heart, which is called the pericardium. It's going to be the outer layer of the lungs or the pleura. It's going to be there's like, it's weird to think about this, but there's space in your abdomen between like your intestines and like everything else, and then your skin, and the space in between is called the peritoneum. So mesoderm is going to form that. It's going to form your cartilage, your muscles, your tendons, all of those sort of like, I just like calling them like the middle linings. That's how I remember like mesoderm middle. And then the outermost layer, of course, we're going to look at the ectoderm. Ecto, outer. So that's going to become our teeth, our hair, nails, skin. It's going to become our neural tissue. Remember that. And that is going to be oh yeah, importantly, sorry, back to the mesoderm. The ectoderm is important for us because we're going to be talking about the central nervous system. and that is going to arise from the ectoderm. But an important part of the nervous system, the early nervous system, is the notochord, which is going to actually be derived from mesoderm. And the reason why that's important is because the notochord is what secretes Sonic Hedgehog. It is so weird to say that sentence. Secretes Sonic Hedgehog. All right, so let's look at another slice of the embryo. This is about 1 month. And you can see very, very clearly at the top here. Ooh. That is the neural tube. Did I label it? Yes, thank God. This is the neural tube is where eventually it's going to sort of be replaced by actual nervous tissue, and it's where our vertebral column is gonna stack itself. And then that notochord that I was talking about is right above it. I know it looks like it's below it here, but the notochord, you're gonna hear this a lot, dorsal and ventral. Just think of ventral as as belly side, as the front side, and then dorsal is gonna be your back side. So the notochord is in front of the neural tube. And you can see everything, all the crap that's in front of this, we're gonna get, they're gonna become other structures. So we have the foregut here. I think we have the respiratory primordium here. And a lot of what's in front of that is just gonna become the heart. So, and you can see by the bottom, it's telling you where the slice is. That it's really important for us to remember that the notochord is in front of the neural tube. So if we go to another slice that's just slightly far, the same embryo, but just slightly farther down, we can see other things that are in the front of our embryo. So this right here, it's facing us, so this is going to be the left umbilical vein. This is the right umbilical vein, and then down here, I think around this area is going to be the umbilical artery. So those are just to give us some frame of reference of like we're looking at the front of the embryo. Very cool and very gross looking. So we're going to skip ahead again, and we're going to go to the embryo at day 56. This is the last day of embryonic development. Hey, Star. Ooh, I like a dorsal fin too. Oh yeah. We love those curly orca dorsal fins. That is honestly how, when I was like studying this stuff, how I would remember. Because it's hard to remember the body axes. It's like dorsal, ventral, anterior, posterior, which are different. Cranial, caudal, distal, proximal. I just think about orca whales. Okay, so day 56, this is the last day that a developing human is considered an embryo. After that, that's when we start calling it a fetus. But you can see that we have a lot more like, yeah, oh, you mean what's it called? Sagittal. Yeah, sagittal is like, yeah. I hate that. Coronal. Stop it. These are actually, I think technically these are coronal cuts that we're looking at. Because we're looking down from the head. Anyway. We see some more like obvious structures here. Right here. This is a vertebra. Right here. This is it was the neural tube in the other slices, but now it's kind of starting to become like, you know, the spinal cord, the spinal column. We have, this is the sternum right here. Oh, right over here. Up, that little thing. This is the chest. We have like chest muscle right there, and then we have a straight up hand. So things are really starting to take shape. And Sonic Hedgehog has really important functions in making sure that the right cells are being developed in the right places at the right time. I mean, you're gonna see that's wow, is that important? And of course the left-right axis too. We have a hand on each side that are mirror images of each other. We have feet that are mirror images of each other. We have 2 hemispheres in our brain that are mirror images of each other. Oh, remember that shit. Oh, that's gonna come up. So yeah, and at this point, remember this section over here, which was the neural tube. At this point, at day 56, the cells have differentiated to the point where it's distinct neural tissue that transmits information and is starting to like take shape and have their own individual functions. Whereas when it was called the neural tube, it was just kind of like undifferentiated. Speaking of differentiated and undifferentiated and all of the weird words that I've said so far, the next thing we're going to go over is some of the development vocabulary that you're probably going to hear me say a million times, and that is going to help us understand, again, what the hell does Sonic Hedgehog do? So the first one is a noun that is now a verb, patterning, or pattern formation. This is the organizing and the induction of the development of specific populations of cells, such that they are being induced in specific areas, and they will eventually become distinct structures based on where they're developing. So we say that Sonic Hedgehog has its main function in pattern formation. Pattern formation, it has its main function patterning the spinal cord, I should say. This concept goes hand in hand with a process called morphogenesis. Oh yeah, and these are the this is sort of the pattern formation when it comes to the central nervous system, which starting with the ectoderm differentiates to become these specific populations of cells that arise and become specific parts of the central nervous system. We'll mostly be talking about the spinal cord, but you know, where does that cord lead to? The morphogenesis is more specific. It's the actual creation of the tissues and the organs that organize themselves into distinct anatomies and systems within an organism. And that's where, when we call Sonic Hedgehog a morphogen, we just like making up words. It's referring to its role in morphogenesis. So these two concepts are kind of similar. How I see it is patterning is something that precedes true morphogenesis, where you have the patterns of cells and where they go, and eventually they're going to morph into the heart and the lungs and the spinal cord. Oh, thank you. Thank you, Gray. Oh my god. This this is the process by which organisms take their shape, and it's really nuts, because there's crazy networks of proteins and genes that are all working to coordinate things and make sure that everything is done correctly. And it's different types of proteins too. It's adhesion molecules, it's receptors, it's transcription factors. All of these things have to be so tightly controlled in a developing embryo. One Sonic Hedgehog basically will tell these embryonic cells what they're destined to be in a way that is nothing short of weird and complicated and crazy. But we'll get into that in a sec. Because I've used the word differentiation a million times. Differentiation is the process by which a stem cell becomes more specific, more specialized, more what it is, basically. And of course, when it comes to embryonic development, stem cells are kind of a hot topic. I think that's probably a term that many of us have heard before. So I will tell you just the true definition of a stem cell is it is it's true that they have the potential to become pretty much anything based on their potency. But another important aspect of stem cells is that they're self-renewing. That's like a really big definition of a stem cell. If a cell can like kind of differentiate into whatever, it's not a stem cell if it can't self-renew. And the big ticket stem cells that everyone just wants to isolate are going to be those totipotent stem cells. They can become anything. They can become anything, any cell in your body, plus anything that's extra embryonic, which remember is going to be the placenta and the amnion and stuff like that. Totipotent can become literally anything. Pluripotent is stem cells that can become pretty much any of the 3 germ layers, and therefore whatever those germ layers could potentially differentiate into, but not the extra embryonic placenta whatever stuff. So, oh yeah, the picture that I have there on the left, the black and white, totipotent stem cells. I can't believe I almost forgot to say this. Totipotent stem cells that can become literally anything only exist during like the 3rd day of embryonic development. That's it. After that, no, they're not totipotent. So what's pictured there is it's called a morula. So that is a human embryo at the 16 cell stage. Those are totipotent. They can become any type of human cell ever. Not even day 4. Nope, because by day 4, that's when we're dividing a little more, and those distinct populations are separating between extra embryonic and embryonic, and therefore they are pluripotent. The last type that I'm going to go over is multipotent, and these are cells that can become a lot of different types of cells, but it's predetermined what those cells are. So the chart I have here, hematopoiesis, this is what's taking place in our bone marrow right now, and it's where all of our blood cells are made. So at the top with the hematopoietic stem cell, that is a multipotent cell. It is going to become these 2 different progenitors, and then those progenitors are going to become a specific subset of blood cells, but not bone cells, not epithelial, not like any other cell. So that's multipotent. So these sorts of processes and words that I've outlined are all coordinated by upregulating and downregulating different genes and therefore different proteins. And these changes are what determine the fate of a cell, what it's going to become eventually. They determine the cell migration, where it's going to go. And they determine the proliferation as well, if we're going to make a lot of these cells or a little bit of these cells. Very tightly controlled, like I said. And at the center of that whole operation is Sonic Hedgehog. I mean, that's going to be our ligand, who sends signals into these different cells and tells them, like, you're going to be this population of cell, you're going to be this population of cell. And the different concentrations of Sonic Hedgehog are going to be reflected by the concentration of transcription factors within that cell. That's going to be really, really, really important. So, everyone just, like, a round of applause. Let's welcome Sonic Hedgehog, the star of Hedgehog Signaling. Hedgehog Signaling. That's what it's called, goddammit. I love it. I sound like I hate it, but I love it. So, Hedgehog Signaling. I like all these guys. They're all so useful. No. Hedgehog signaling is a highly conserved pathway, basically. The hedgehog signaling pathway is highly conserved, meaning that it happens in pretty much every single living thing on Earth. And I mean, it truly connects us humans to other animals that utilize the same pathway, to our special Drosophila, our mice, and our Xenopus, our frogs. Xenopus is actually like huge in studying development because their eggs look so cool. Basically, their zygotes look so cool. Anyway, I want you to keep 2 things in mind as we go through this, though. One is that our understanding of how this pathway works comes from experiments done mainly in these 3 animals, plus zebrafish and sea urchins. But, you know, not human. It's just, it's so much easier to do the experiments in these animals and to figure out which genes and proteins are involved, and then just try to find the homologous ones in humans, exactly like we did with Sonic Hedgehog. And we will when we do it on mice, especially because they're also mammals just like us, we'll sort of just like copy paste and be like, yeah, that's probably what happens in us. So, you know, it's not that bad because the processes are very similar. But still, we're humans, we're different. So just keep that in mind. Yes. Yes, actually. I want to say plants too. Oh my god. I mean, I haven't, I actually don't know because in all of my studies there wasn't mention of plants, but that would be a really easy like way to look at this. I'm looking it up right now. I'm so Nope, not I didn't even it's giving me Sonic the Hedgehog. Oops, I forgot to put protein. Okay. Oof. It looks like it might not be in plants. Yeah, interesting. It is still, I would still consider it like, it's still highly conserved. Maybe not ubiquitous, you know, those are the different levels. If it were ubiquitous, plants would probably have it too. But, oh my god, there's a living plant girl. I didn't look that hard. Is she pink like Amy? Because I just saw like an Amy looking character before I like fixed my search. um, that's weird. I guess plants, or it might just be called something else. Like, a lot of my research on this was very like, I was calling it, oh no, I'm calling it the fly gene this whole time, but I want to call it the human gene. So maybe there is like a Sonic Hedgehog homolog in plants, but either we don't know it or we call it something completely different. So my cursory search didn't come up with it, but what a great question. What a great point. That would be really interesting. I want to look more into that. The other thing to keep in mind is just that a lot of experiments were done in vitro, in, you know, petri dishes and test tubes. So not all of these experiments were done in vivo, in like an actual like living organism. I mean, plants are good for, I don't know, certain things, but when it comes to genetic experiments and development, not good at all. They don't have a nervous system. They don't have the organs that we have. In fact, their way of developing and reproducing is not anywhere near how humans or even mammals or even flies do their thing. I still thought it was possible that there could be like, you know, things that, you know, signal sort of growth stuff, but I guess plants have their own stuff. But yeah. So yeah, that's the last thing to keep in mind is some of these experiments may not have been done in an actual organism. They could have just been done, you know, in a tube of some sort. But we've still learned a great deal from those experiments. They're still really, really important. So I just wanted you to keep in mind that not all of these pathways were understood in like a single like actual living organism, maybe. But we do know it exists, so we're gonna see what it's all about, and we're gonna go to our embryo at day 19. 29. 19 is gastrulation. This is 29. So we can see a very, very defined neural tube in the back, and we can see the notochord on the bottom. What's gonna happen around this time is the notochord is actually gonna start releasing Sonic Hedgehog. and it's going to release it into the ventral area of the neural tube and induce the production of something called a floor plate. The floor plate is at the floor of the neural tube. It is just a distinct population of cells that we call the floor plate. So consequently, the floor plate, which is the orange right there, the circle is the notochord. The floor plate then, after it gets Sonic Hedgehog from the notochord, it is going to release its own Sonic Hedgehog into the cells of the developing neural tube. The Sonic Hedgehog that's being released in those areas is then going to, of course, tell the cells what they need to differentiate into. And it's going to do that through its gradient that it's going to create. It's the Sonic Hedgehog gradient that is going to help define the boundaries between the different cells. So we'll go into the actual pathway now, and we're going to start with the two proteins, patched and smoothened. More great names. We love it. So right now, in this, there's no Sonic Hedgehog. These proteins are just hanging out. Right now, patched is actually inhibiting smoothened. It is preventing that protein from going and performing its activities. When Sonic Hedgehog binds, though, that inhibition is going to be lifted. And the inhibition on smoothened is going to be lifted, possibly through a mechanism involving cholesterol. See, this is where we want to know so bad, but we just don't. This is just interesting. We think that cholesterol is what activates smoothened, like having cholesterol bind to it is what releases it from jail and lets it go do its activity. And so it's possible that patched prevents cholesterol from coming into the cell at all. And then when Sonic Hedgehog binds, it sort of opens a channel that allows it into the cell. Molecular. It's very molecular. It's very specific. But during this process, patch is actually downregulating Sonic Hedgehog because so smoothened gets released from jail, basically. The receptor is going to bring Sonic Hedgehog into the cell, endocytosis. And by doing that, it's downregulating Sonic Hedgehog. I mean, this molecule that's bound to patch right now, it's not going to go out and activate more things. It's just, its job is done. So that is a form of, that's a form of negative feedback. The pathway is really long and complicated, and I'm taking parts out that I don't need to say. Sorry. Okay, here we go. So, as a result, like smoothened gets released from jail, and as the gradient is continuing and more Sonic Hedgehog proteins are activating patched, we're gonna accumulate some smoothened within the cell. Specifically in the primary cilia, which I didn't draw, but I wanted to mention anyway, because apparently it's really, really important that this is happening in the primary cilia of the cell. So the cilia is just like the kind of fingery structures on cells. It just is important. I couldn't get much more than that. It's just important. What smoothened is going to do then is it got released from jail, right? So now it's going to release some other bitches from jail. This complex of proteins right here is holding a transcription factor hostage, the Glee transcription factor. In our hedgehog signaling, we want that out. We want that to be doing its thing. But right now, it is being tethered to the microtubules in the cell by a protein called KIF7. And it is being prevented from going to the nucleus, which is where you want your transcription factors to be doing their job, by a protein called SUFU. Now, SUFU prevents Glee from doing its job in two ways. One is, of course, it physically prevents it from going to the nucleus. And two, it actually degrades it into GLI-R, which is a repressor. So repressors are transcription factors that prevent transcription. So when we have GLI bound to SUFU like that, we don't have a lot of the activating form of that transcription factor. But when smoothened is increased in concentration, thanks to Sonic Hedgehog, it's going to release it from SUFU. So when that happens, it's going to be, we're going to get a higher concentration of that activating form of GLI, and it's going to then go do its thing in the nucleus, which is upregulating other proteins that are going to officially define our cell boundaries in our developing neural tube. So I'm going to backtrack really quick, actually. Oh, I did write it right here. When there's no hedgehog, patched is inhibiting smoothened. No transcription, basically, is happening. Transcription is being repressed. With the activation of the hedgehog signaling pathway with Sonic Hedgehog binding patched, it gets taken into the cell, negative feedback. Smoothened gets released, and then smoothened releases another transcription factor from jail. So this is going to be a better visual on that, because I know that sounds really, really weird, but really what's happening is the gradient of Sonic Hedgehog, where you have a lot more Sonic Hedgehog at the ventral parts of the neural tube, the front parts of the neural tube, that are progressively getting lower, 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 lower in concentration as we move along. That same thing is happening with GLI as it's getting activated by the Sonic Hedgehog pathway. So we have lots of this transcription factor activity in these areas of the neural tube, and then we get progressively less and less and less. And just what these red ones are showing is these are the repressive forms. So because no Sonic Hedgehog means that we have the repressive form of GLI, these back parts of the neural tube that don't have a lot of Sonic Hedgehog are going to have a high amount of repressor. So this visual is really good. This like really summarizes everything and like breaks it down. So, but again, GLI is still kind of a middleman, because what those GLI proteins are going to do is they're going to activate these other types of proteins called Homeobox proteins. Homeobox proteins. Those specific types of proteins have to do with body planning. And they have the actual direct effect on the patterning and morphogenesis of the neural tube. And we discovered them in flies, of course, because when there was a mutation in one of the Homeobox genes, it made it so that the flies, instead of having antennae on their head, they had legs. So those are supposed to be antennae, but those are legs. That fly is like high-fiving itself with its leg, not antenna. And experiments with the Homeobox genes are always really interesting, because if you take them and you're like, I'm going to express this over here, that's how you can get like incorrect body parts growing from like weird areas. So those need to really be expressed in the right place and also not be mutated. We don't want legs on our heads. Like we really, really, really want those boundaries to be fixed. So we are going to take a look at the boundaries about, I think this is gonna be a month. This is a month into, this is day 30 of embryonic development. Again, you know where it is. You know the neural tube. It's the very vaginal looking thing. I'll just say it. We'll just say it. It's the very vaginal looking thing. And by this point, the notochord has released Sonic Hedgehog into the floor plate, and it's creating this gradient. If you don't know by now, what I mean by gradient is basically just that there's a lot of Sonic Hedgehog down here, and it just gets progressively less and less and less until we reach the dorsal boundary. because there's something else up here that is doing its own thing similar to Sonic Hedgehog, but with the dorsal guides, which we don't care about. We only want the front. We only want the ventral. So the reason why this this gradient happens and is maintained, the gradient, it's not like eventually there's going to be like a lot of Sonic Hedgehog in these areas as time goes on. No. The gradient is being maintained because remember, that receptor patched is negative feedbacking our Sonic Hedgehog. And as it like moves across this gradient, it's upregulating the thing that's eventually gonna take it back into the cell. Because this is the only place down here, this is the only place that's actually releasing Sonic Hedgehog. So these areas get to stay high, these areas get to stay low. And the cells of the neural tube in this area are multipotent. So they can't become anything, but they're going to become a specific subset of neurons or glia. Glia are cells of the nervous system that provide support and stuff, but they don't conduct nerve signals like neurons do. So as Sonic Hedgehog is traveling along the neural tube in these cells, these populations of stem cells will be induced to become slightly more specific. They'll become progenitor cells. Progenitors just being like, I still kind of see them as like multipotent cells, but they're not stem cells because they don't self-renew, but they become a specific subset of cells. That's progenitors. They have specific domains within the neural tube, and this is this is what they are. These are the nervous tissue domains within the neural tube. So don't worry. The colors will guide us to victory. On the bottom, always have the floor plate. Love the floor plate. Releases Sonic Hedgehog. And because the belly side is where we're going to have the most Sonic Hedgehog, I just really want to always drill that in. Most Sonic Hedgehog at the ventral side, not a lot of Sonic Hedgehog at the dorsal side. That's the most important thing to remember. The moving backward from the floor plate, we have our first domain, the P3, progenitor 3 domain. This area is going to give rise to V3 interneurons. Interneurons are our middleman neurons. They take signals from sensory neurons and they relay them to motor neurons. The ones that we're going to be talking about are pretty much all in the spinal cord, so they're important for reflexes. They just relay information really quickly, but they're not motor or sensory neurons. And a lot of these domains are going to be interneurons, and the thing that makes them different is where they are, the neurotransmitters that they express, and where they go. So the next one we have, this is going to be our motor neuron domain, and it actually divides into both motor neurons and oligodendrocytes. This is the area where Sonic Hedgehog apparently has the most influence. Anyway, oligodendrocytes are glial cells, they're support cells that add myelin to our nerves. Super important. It's like this fatty substance that increases the conductivity of our nerves so that information travels really, really fast. So very important. The next domain is going to be the P2, V2 interneurons and some astrocytes. Then we got the P1 going to the V1 interneurons and some astrocytes. And then the V0 interneurons. So lots of interneurons because that's a lot of what we have in our spinal cord. I think I could give an easy example when it comes to the reflexes. So the doctor does that thing where they hit your knee and you kick, right? That reflex involves extensor muscles being activated. You are extending your leg. So you have interneurons in your spine that are preventing you from flexing them, from flexing the flexor muscles, which what's it called? It does the opposite of kicking out your leg. It inhibits the muscles that bring your leg in. Something that you didn't know you needed, right? Like, really weird concept that our interneurons in our spinal cord would just inhibit something like that, because it's like, I know I'm kicking out, but you know, that's just a good example of what interneurons are doing when it comes to the reflex arc. They're very automatic, quick reactions. How these okay, I did include how the interneurons look in our spinal cord, because I know this was probably really hard to visualize with just the vagina looking thing, but this is the front and this is the back of our spinal cord. So what was that first domain? P3? Boom, V3 is right here. It's the frontmost one, followed by there's two populations of V2, V1 over here. So they're getting sequentially induced. And then they eventually in development will have their little outcroppings, their axons that go up and down, or just up. But this was just to visualize in like the extending neural tube, three-dimensionally, like where these guys are. And it really seems like these guys are similar, and they are, but they do have distinct functions. There are reasons why V3 is induced closer to the front of the neural tube than the other guys. Like, there's a reason for that. And things just need to be where they're going to be. So there are quite a few cell types, as you can see, coming from this area. And having these boundaries defined is all thanks to Sonic Hedgehog and the proteins that it's going to induce. And it probably won't be till later that I impress upon you. It is so important to have these boundaries. It is so important. You don't want motor neurons being induced in this area and vice versa. Like it's hard to like really feel how important that is. Oh, those are cute. That unicorn does look like a dick, though, I gotta say, but I like it. I like it. It looks like a dick with a unicorn horn. Oh my god. Okay. Well, is there a nicer slide than this? Well, no, these are all brain hurting. Dick unicorn, yes, it's the best. The next slide's gonna be rough, I'm just telling you right now. Because we're gonna look at the wow. Homeobox genes. We're gonna look at those Homeobox genes that I was telling you about, and like where they're expressed in these specific areas. Bless you, Amy. Okay. Here we go. Boom. Don't worry. It's okay. Don't worry. The names are stupid. They look weird. We don't care about the names. What I want to tell you right off the bat is on the right side is those domains that I already drew out in the neural tube. So we've seen that before. I really just want you to worry about this. Just look at this. It's okay. They're really stupid sometimes. Like, don't look here at all. No, you're not stupid and weird. Well, you're weird, but not stupid. No, I love your name. I think you have a beautiful name. And I don't think you look weird. I think you look stunning. So, anyone who thinks that can go frig off. But I showed you the progenitor domains, so that's familiar, okay? We got the floor plate on the bottom and the progenitors. It's good. On the left, this is just showing you through which domains each homeoprotein is being expressed. So the reason why they're separate is so that we can visualize the boundaries a little better, which I will take you through. Homeobussy. My homeogenes patterned my homeobussy very correctly. But you can imagine these as being all just on top of each other, like all on top of each other. But we're gonna have them split so that we can visualize it a little better. So these genes, these Homeobox genes, when it comes to Sonic Hedgehog, we divide them into two classes. We got our class, oh yes, oh my god, how could I forget? The sequential activation of Homeobox genes along the ventral neural tube defines the boundaries between the progenitor populations, which I'm about to demonstrate. So the two classes, though, we have class 1, repressed by Sonic Hedgehog. Class 1, Homeobox genes, are repressed by Sonic Hedgehog. Class 2 Homeobox genes are enhanced, increased by Sonic Hedgehog. And as I take you through where these proteins are expressed, I'm going to show whether they are class 1 or class 2. Okay. And we'll back up for a second. We're just gonna chill for a second. This is some molecular shit. This is some like grotesquely existential kind of shit here. So again, I really like. We don't need just think about the colors. You know, don't even think about the names. The names are dumb. We don't like the names. There's too many X's. There's too many decimals. We don't like it. The important thing that I'm going to be demonstrating here is that gradient of Sonic Hedgehog that is being maintained throughout the ventral neural tube is recruiting transcription factors called GLE, which are mirroring same gradient, but inside the cell. And those two gradients are what is going to create these very specific expression patterns of these transcription factors that are going to make sure that the correct cells arise in the right areas. Glee. Oh my god. Jane Lynch burst down my door. Oh my god. All right, here we go. And again, just remember the left side, you could superimpose them on top of each other. It's like throughout, they're separated for visual help thing. But we're the first domain is that P3 domain in red, which borders the floor plate and the PMN. In that area, we see definitely two transcription factors that are expressed there. NKX 6.1 and NKX 2.2. Both of those are in the P3 domain. They are both also class 2 proteins. However, 2.2 does not extend past that P3 boundary. And it also doesn't go down into the floor plate either. NKX 6.1 is totally allowed to do that, though. So what's the big idea there? Well, at that P3 domain, there is another transcription factor called PAX6. Now, PAX6 is a class 1, and it represses 2.2 at that boundary. In fact, they repress each other. So PAX6 can't extend past that boundary, and then 2.2 can't go up past that boundary. So part of what makes that boundary is the fact that those two transcription factors are mutually repressing each other. And we got a lot of Sonic Hedgehog in that area, right? And that's a class 1 protein, which is repressed by Sonic Hedgehog. So additionally, PAX6 is being repressed by Sonic Hedgehog itself. So that's what makes that rigid boundary between those areas. The next area that we have is OLIG2. which is another class 1. No. I almost ruined everything. It's a class 2. Whew. That is induced by very, very, very high glee activity. So the levels that are in the P3 domain, though, are really, really high. And even though OLIG2 is repressed by that really high activity, the crazy, crazy high amount is going to induce the 2.2. And guess what? Those guys mutually repress each other. So even though they both are being induced in the same area, they repress each other to create that boundary. So moving down, we see in the boundary between PMN and P2 that IRX3 is being expressed. It is also, it's going to be a class 1, just like PAC6. It's another one that's going to repress OLIG2. So in this area, we could see, like, just think about OLIG2 for a sec. It's being repressed at this area, and it's being repressed up here. So 2.2 is defining the ventral boundary of OLIG2. IRX3 is defining the dorsal boundary of OLIG2. These boundary definitions are really important. So the next domain, of course, is going to be the P1 domain. So we have another NKX 6.2. It's a class 2. It gets repressed by the class 1 DBX1. And then lastly, at our last boundary, or second to last, sorry, we have finally something repressing the 6.1, the DBX2, which is a class 1. PAC7 is a class 1 that defines the dorsal limit of the whole Sonic Hedgehog gradient. And it's important to note that throughout this gradient, the cells in the very back that are farthest away from Sonic Hedgehog production are going to be the most sensitive to Sonic Hedgehog because they don't have as much patched proteins there. So it's not going to take very much for them to react. So even though there's not a lot of Sonic Hedgehog in those very back boundaries where we have PAC7 being induced, that's okay because they're sensitized. Only a little bit of it is going to recruit the glee repressors and repress PAC7. We don't want PAC7 in this area at all. It is a dorsal homeobox protein. We only want it in the back, not the front. Jesus Christ. That was a lot of molecular biology. Let's look at the triangles again. Just to visualize. Because it is very interesting that all those class 1 proteins are located dorsally and all the class 2 are located ventrally. And if I visualize this gradient right here, it makes a lot more sense. We have more Sonic Hedgehog in the more ventral areas, so we want those class 2 ones to be upregulated. And if there are any class 1 that are there, good, we'll have them be repressed. We only want them in the dorsal areas. And I know that it's probably really difficult to imagine, like, I don't know, because we didn't name these transcription factors anything, or these homeobox proteins anything like, you know, interneuron creator 1 or like motor neuron creator 8. Like, they're just, what are these names? But as you can see, the gradient created by Sonic Hedgehog here is making sure that transcription factors that we need in the more dorsal areas are not being induced up here. And the ones that we that are more ventral that we don't want in the dorsal area are only being induced in the more ventral areas. Sonic Hedgehog orchestrates this. Sonic Hedgehog is like, yes, I want NKX 2.2 to only be in the P3 domain. Ha ha. And then DBX1, you stay back and don't come any closer. You stay in the P0 domain. I mean, wow. This is so goddamn cool. It's so cool. I wish that we as like a people understood more about what these Homeobox genes did specifically, because I did, of course, I did a lot of research, but there wasn't any cohesive description on like, what does NKX 6.1 do? Oh, it's in the floor plate to P2 progenitor domain. Well, that's where it is, but what does it do? Like, it's really hard to figure out exactly, like, function-wise what's going on. In fact, these results came from, groups of scientists expressing specific genes and staining them so that we could visualize where they are and seeing like, oh, this one's here, and this one's here. Why is 2.2 needed for P3? We don't know, but it's there, and we know it's there. I wish I could go farther and explain to y'all why it's so important that these specific, homeobox proteins are in these areas, but we haven't gotten that far yet. But it's just like, it hurts physically to think about, like, oh yes, Sonic Hedgehog, a bunch of those particles are just making other particles being released inside the cell, and then those particles inside the cell are making these other particles. Pisses me off. All right, with the domains, just one last thing. It's okay. I just wanted to show why this gradient gets maintained. Why it's not like, oh, eventually you have a lot of SHH up there. No. So, let's say, I think this is supposed to be the P3 domain. So it's the first one after the floor plate. You got lots of glee activity. Wow, you got so much. And this is the first, because we're at time 0. This is very early. We got a lot. This gets induced first. Okay, that's interesting. And as we keep going, now we have even more Sonic Hedgehog, and it's going farther. And we have even more. You see right here, the orange thing is patch. We got more patch, so we got more of the negative feedback happening. And so now, oh, we got Olig 2 in these guys now, but it hasn't yet reached here because we're just not, we got too many patched proteins right here doing the negative feedback thing. Even more time passes. Oh my god, we have so much Sonic Hedgehog and therefore so much glee that Olig 2 actually gets repressed in this first cell, and it gets induced up here, but not up here because there's not enough glee yet. Because negative feedback. Okay? Negative fucking feedback. Oh, and what the hell's going on the opposite, more dorsal side of this cell? We got lots of repressor activity. And we like that for PAX7. PAX7 is a class 1 protein. It's repressed by Sonic Hedgehog. So when there's no Sonic Hedgehog, it gets to be existing. But because these cells, like I said earlier, are very, very sensitive, only like a little tiny bit of Sonic Hedgehog is enough to repress it. And now it does not exist in this cell, and a boundary is created. Negative feedback. You get repressed and you make boundaries. Oh, you're sensitive. Oh my god. You're just so sensitized to all the stimuli in your life. It's not your fault. The negative feedback mechanisms that some people make over time just, haven't been created in yourself yet. It's okay. I'm also very sensitive sometimes. I'm a class 2. I'm a class 2 Sonic Hedgehog protein. Oh my god. Okay. So in this last area, the Sonic Hedgehog does not reach up here because the gradient, all of these things beforehand, are acting to prevent it from reaching the dorsal limit. Ugh! You guys! Oh my god, this is why you have motor neurons in the right place! Oh, this is why we have glial cells in the right places. Oh my god! And this is just in the spinal cord. I just want you to remember that it's a tube, right? And it goes like up and it goes up, and then what happens? Oh, it's your brain now. This is going on in your brain during development. More on that in a little tiny bit. I'm okay. Everything's fine. The last thing I want to say before I go into like the kind of other things that it does is do I have a visual? Okay, I do. This guy again. So you see how these I'm going to circle the populations of cells that are induced by Sonic Hedgehog. Do you see how they kind of, oh, like this one goes down and then it goes this way and this one goes down and then it goes this way? Oh, not you, actually. Wait. Not this one. I was trying to do this one, but I can't see it. This one. This goes down and it goes this way. Sonic Hedgehog also plays a role in Axon Guidance. So what we have right here, this is an axon. It's an one end of a nerve cell, and because these are interneurons, they're gonna synapse with other nerves and create, relationships. And something that Sonic Hedgehog does is these cells. These are the Sonic Hedgehog ones, but also these guys, the ones that are coming more dorsally. There is a protein called BMP7 that tells these to make axons go this way, toward where there's a lot of Sonic Hedgehog in the ventral neural tube. Once they reach that really high concentration of Sonic Hedgehog, because during this time there's low Sonic Hedgehog in here, like in the dorsal area, right? But as it starts creeping, the Sonic Hedgehog proteins will guide it and be like, yes, come this way, come more ventrally. And once they do that and they get to really high Sonic Hedgehog concentration, they just go one way or the other. They guide themselves. Once they reach the midline, they go in other directions. And we think that Sonic Hedgehog plays a role not just in like, yes, come this way, axons of the dorsal neurons, but, and this is just my thought because I couldn't find definitive answer to this, so this is my thought, that really high Sonic Hedgehog concentration at the very front is what eventually repels it. I'm not sure what makes it go up or down or like tells the axon like which way to actually go, but Sonic Hedgehog still does have a role in guiding the axons toward that ventral midline before they go off and, marry or whatever. So that's kind of cool. There are others. I think like lung development involves Sonic Hedgehog and stuff like that, but another one that like we've studied kind of is limb development. This is a mouse. The bottom one, the digit 5, that is the pinky. It has been observed that lots of Sonic Hedgehog are required for the pinky differentiation into telling these cells that you are the pinky, and then it gets lower and lower and lower until we get to the thumb, which is, I think, completely independent of Sonic Hedgehog development. You are pinky. You have many Sonic Hedgehog. Like, it's just another area where we see the Sonic Hedgehog gradient at work, where in this case it's Anterior, posterior. I feel like. Okay, fine. It's moving along that posterior anterior axis. So in another way, it's saying like where, like it's having a role in body orientation basically. So, pretty interesting. Not as well studied, but just interesting. I don't know that we've seen this in humans yet. I think it's really just in mice, but yeah. Anyway, who is ready for some fucked up stuff? Who's ready for some fucked up stuff? The next slide is not body horror. It's just a little bit in a couple slides. I'm gonna have another one before I'm actually showing some things, but there's gonna be some descriptions and I'm gonna be talking about it. So the warning is here. Yes, it's food safe. It is food safe. I didn't I'll just say right now, I didn't include photos of humans. There is a drawing, but it's very like it's a drawing. I'm like fairly certain that this is it's food safe. You got it. So patterning the central nervous system is really important, right? And you can imagine that some pretty screwed up things happen if it doesn't get done correctly. And we were hanging out in a pretty deep part of the neural tube. But like I said, that tube goes up and eventually is the brain. So this is a head-on view of the early embryo brain on the left. It even, I mean, it kind of looks like a freaky embryo, right? So this is a side view, and we're going to be mainly focused on the prosencephalon, which is going to become our forebrain. I think it's about 35 days in. The forebrain, the prosencephalon, will split into the telencephalon, which will eventually become the cerebrum, which is like the meat and potatoes of our brain, basically. It's like the brain. The olfactory bulb and the hippocampus. The other section it will split into is the diencephalon, which will be our thalamus and our hypothalamus, which are important for like homeostatic regulation and integrating signals, and the optic vesicle, which is our one of our points of interest here. So that's 35 days in. That importantly, our forebrain becomes those distinct areas. And let's take a look at this is day 56, so this is the last day of being an embryo. This is when the prosencephalon splits into the right and left hemispheres. Really important, as we're gonna see. We have the right and left hemispheres of our brain, right? Like we need that. You can't have one hemisphere. And we can see, where's my pen? We can see mirrored structures, right? This right here, this area this is the basal ganglia. Haha, our old friend, the basal ganglia. It is mirrored over here. So in this particular embryo, fetus, kind of both, because it's day 56, we have definitive evidence here that the brain has split into the distinct hemispheres, which is what we want. And this next slice is the same time, same embryo. I just wanted to show the where the optic vesicle is going to be. So we got two halves of our brains, and we have two eyes. That is not by accident, and they 100% have to do with each other. In fact, a lot more intertwined than I thought, but the development of our eyes is kind of dependent on this splitting into the two hemispheres. And not surprisingly, Sonic Hedgehog is gonna help out by recruiting specific transcription factors in the areas and making sure that the correct progenitor cells become the correct areas of the eye, basically. There's a lot going on there. There's a lot. There's much like what I've already described, those processes that I've already shown you in the spinal cord. That's not everything, you know? Those are just like the big ticket items. There's a lot more going on. And especially in the brain. There's a fuck ton of shit that's happening up there. Even in regards to Sonic Hedgehog. Eyeballs, ice balls. Amy always says eyes balls. My butt's hole. We said it at the same time. Anyway, this time I'm only gonna we're gonna do 2 transcription factors, 2 homeobox proteins. Okay, I promise, just 2. This visual that I've been showing you, I mean, it's wonderful, right? I only drew the progenitor domains on one side because it was easier, you know, but it's mirrored on the other side of the neural tube. It's mirrored. Just wanted to like make sure that we knew that. It is equal exact things are happening on the opposite side of the neural tube. This it's a categorization of humans and a lot of other animals, but we are bilaterian. We're bi ladies, okay? That means that we have mirrored left and right, you know, hand, both side, arm, eyes, nostril, ear, bilaterian. Our embryos are, they have that axis. I try, you know. I try to be very hashtag relatable. It comforts the people around me, and that's always my goal. So having, of course, like, this isn't actually kind of what happens, but like our body makes splits in certain directions. And it's like, oh, arm on this side, arm on this side. Of course, that's going to happen in our brain. The neural tube goes up to our brain, and it's like, oh, we got this side and this side. Perfectly natural. The way that Sonic Hedgehog is going to pattern our ventral neural tube in the brain, as I'm going to show you, is I'm going to simplify it a lot more. And we're just going to pay attention to 2 sections. So the more ventral area is the optic stalk. It's going to become the optic nerve. It's controlled by PAX2. PAX2, at that boundary between the yellow and the green, represses the gene that controls that area, PAX6. That area is supposed to arise and become the optic cup, which will eventually become the retina, iris, and ciliary body. So, like, other parts of the eye. And that, like, sounds... I think the next slide has a visual, because the fact that this is ventral and the optic stalk, and the optic cup is dorsal, like, doesn't make sense to me. But we'll see. We'll see a head. We'll see a brain. Anyway, it's important that these populations stay where they are, and that those homeobox genes repress each other. We don't... This is our eyes, for God's sake. We don't want any mixing between these 2 progenitor areas. That would be bad. Okay, yeah, this is good. So we're kind of we're gonna see what happens when they mix. Because the thing is, the splitting of the forebrain into the distinct halves, in order for our eyes to be correct, that must happen. If they are in the wrong areas, our eyes will be really close together, or maybe even like really far apart. There's something called aniridia, which is when you're born without an iris. So your eyes are basically black. That's usually mutations in PAX6 because the optic cup becomes the iris. So if you have mutations in PAX6, boom, you don't got a fucking iris. It's freaky. It's freaky looking. You want that? It would look really, I mean, like, I've seen pictures and it's like, like if someone like, if I was interacting with someone who had aniridia, I might not notice right away. You know what I mean? Like, I would really have to be like, oh my God, you don't have color in your eye. Holy shit, it's just black. It's just black. All right. But this kind of shows from the front view, our eyes are actually, they're facing this way a little bit initially. So that's why, like, the more, I don't know, like, where is it? Does it? Okay, it doesn't actually show it. So this is the stalk right here, the optic stalk, which is gonna, that's where the optic nerve, it's gonna become the optic nerve. And then this right here, this evaginating area is gonna be the optic cup. That's gonna be like, you go around and be the iris and the retina. Anyway, so you need the correct division of the two brains in order for your eye to be eyes and not eye. Spoiler alert. Two halves of the brain, you need them to split. Brain doesn't split, that means your eyes don't form correctly, or you don't form eyes and you get eye instead. Spoiler alert. So what happens when the brain doesn't split like that? It results in a birth defect called holoprosenchephaly. I'm gonna show another trigger warning. It's gonna be an ultrasound first, and like I said, the main body horror, it is gonna be a drawing. I'm not gonna show actual pictures, and I will explain that, but it can be upsetting, so I put the warning. But again, I'm fairly certain it's food safe. Especially this, it's just an ultrasound. So, yeah, so the blue is showing us there's supposed to be, normally it would be kind of split, right? You would have like something going in like that. Were they pregnant in their butt? I mean, this is the transvaginal scan. Ew, Amy. But yeah, there's no lobes. It's called alobar, which means that no lobes, or one lobe. And so there's no fissure down the middle of the brain. This, I believe, is a 12-week embryo. There are other parts of the brain that you can see that are fused when they're not supposed to be. Yeah, Amy's laugh is the best. This is one of the more severe subtypes of holoprosenchephaly. The prognosis is not good. I wanted to find statistics, but they were not. I found so many conflicting ones that I'm just gonna I'm gonna ballpark this, but it is really bad. Pretty much half of fetuses who have holoprosenchephaly like this are stillborn, or they miscarry. And if they are alive when they're born, they probably don't live to even 6 months. It's not a good prognosis. And there are differing levels of this. This is the more extreme one, not the most extreme. I'm going to show you the most extreme in a bit. But having the one lobe is the worst, or like the most serious one. You can have slight, like, there are fetuses that have like slight fissures that are, it's good. They have almost split the brain. But they tend to have, they'll have like facial abnormalities, like usually like some sort of unevenness, or their eyes might be pretty close together. Usually the nose is a little bit high, and cleft palate, like facial abnormalities like that. And then I believe there tends to be certain levels of like intellectual disability. And also other stuff like a lot of them will need like a feeding tube depending on how bad. Like I think depending on the abnormalities in the face, there's trouble eating. Yeah, it's sad. It's yeah, no gray, no gray. Yeah, no, I should have I meant to say that actually. I was going to tell some people like I know some pictures if you want to see. Don't look at any more. I know you probably saw enough, but like don't do it. It's I feel like I feel emotional. Like I saw a lot of these pictures. Um so yeah, I wouldn't do any more. Um so this comes about because of mutations in Sonic Hedgehog itself, like its creation, or maybe it's a messed up, protein itself, or in mutations in any of the proteins in Hedgehog signaling, patched, smoothened. Um, there's a protein called dispatched that helps release Sonic Hedgehog. So mutations in that can also cause issues like this. But there can also, I mean, anything, Glee, Homeobox proteins, anything like that. The most extreme, this is not the most extreme. This is the second most extreme form of holoprosencephaly. The most is it has to do with inhibiting the hedgehog signaling pathway at a specific place, and it was first observed in sheep. So this is like this sheep actually. When I found this picture, I read that the sheep it did not live. It lived like for 2 days. So this is a pretty lethal mutation. But basically in the 50s, there were like farms that had sheep in Idaho, and they were being born like this. They were with the one eye and like the weird faces and stuff like that. And they learned that the sheep were eating something called a corn lily, and the corn lily makes something called cyclopamine. Yes, it is. Yes, it is. Don't look it up. Gray. Don't don't do it. um so what cyclopamine does is it okay, good. Yeah. It's bad. Cyclopamine has a really high affinity for the for patch, for the Sonic Hedgehog receptor, even more than Sonic Hedgehog, like higher affinity than Sonic Hedgehog. I do the same thing all the time where I'm like, I regret looking this up. Um and cyclopamine, it binds patch. Patch brings it into the cell. Cyclopamine binds smoothened, and then it also inhibits smoothened. But there's no like downstream communication about what is inhibiting smoothened. Like with Sonic Hedgehog normal signaling, it's being inhibited in a way that's normal, so the cell is normal. But if it's being inhibited in a weird way by this weird guy, it's not gonna work. So yeah, this is cyclopamine. It's binding patched, and it binds smooth, and it makes it not do anything. And as a result, sheep are born like this. And again, so I'll show you a draw. This is a drawing with a as of a human baby with Cyclopia. It's like a clinical illustration. So it's pretty rough, but it's not gory or anything like that. So that's what's coming up. So these babies, their forebrains did not divide, and because of that, they only developed one eye. And you can see they kind of have two irises, as if like the body knew that it was supposed to, separate, but they have one eye. And then that thing coming out, that's called a proboscis. and it's apparently what the nose is supposed to be. So as our faces are forming, the nose comes out and then it eventually moves down and forms. So the lack of dividing of the hemispheres, I mean, it affects a lot of the development of our faces. So it really clearly affects the development of our nose to the point where it just looks like an uncircumcised penis on the forehead. And dude, yes, when I was taking cell biology and later development, it was actually mostly cell biology, but those classes together really made me feel like there's a lot that can go wrong. There's so much that can go completely wrong that we don't even understand, or like how would we even stop it? Like it's just it's really, it's so scary. I would say more cell bio, because in cell biology we learn about the intricacies of the cell cycle and how cells divide. And I was like, how do we not just like have cancer all over our body 24/7? Like it seems like there's a lot of things in a lot of areas where things can just go wrong and be wrong. And it's really terrifying. Um right? How? I don't know how any of us make it. Like uh if anyone here, by the way, not gray. Not gray. Don't do this, gray. If anyone wants to see actual pictures of holoprosencephaly in humans, um the article, I don't know why, but Wiki's article for proboscis, not the insect one obviously, but they have an article specifically for this outcropping. The article for proboscis has as its main image a human baby born with cyclopia. It has one eye. It is very upsetting. I don't. It's just if you're curious and you can handle that kind of stuff, and you're fascinated by the body messing up. I mean, it's a scientifically it's a very cool picture, but it is not. It is very upsetting. And the reason why I didn't include them is because they're they're stillborn. All the pictures of Cyclopia for humans, all the ones I found are stillborn. Heck yeah, you might have to go like to the disambiguation or whatever, but I actually saw it because on the page for Cyclopia, I moused over proboscis because I was like, oh, there's a name for it, and it shows you the preview, right? And so I got a face full of the image in the preview. It's very disturbing. Yeah. I I thought about showing them some of them for science, but because they are dead, that's what made me think that I would just, if anyone was interested, they can look. Proboscis and Patau syndrome. I'm gonna write it in chat. The wiki page for Patau syndrome has a photo from 1912 of a baby. Still very disturbing. Not as like I think it might be live too. I think it might be alive, so it's not a it's disturbing, but it's like it's a baby. But yeah, I would not look up Cyclopia or proboscis or even Holoprosenchephaly. Like they're not always that bad, but it's very upsetting given that a lot of the clinical photos are of deceased specimens. Yes, I forget the name of that too. It's nasty. There's also this is another Sonic Hedgehog related one, spina bifida, where the neural tube doesn't close all the way. So you just have an open like spinal cord. Like holy shit. It and like I said, this is I'm not trying to say anything about abortion or pro-life or anything like that. I just want to point out that uh this I believe is a 12 week, and you can see defects like this early. It's really easy for doctors to tell that the forebrain has not split up. Oh my god. Yes, I'm gonna need to look that up. Oh my god. We're having soft tacos later. Oh man. Dead babies, am I right? Yeah, that got real serious, didn't it? Uh Mutations in Sonic Hedgehog are fatal usually. So like imagine the doctor coming into your room after 30 hours of labor and being like, "Yeah, so your kid has a poor prognosis because they have a mutation in the Sonic Hedgehog gene." I mean, this was an actual controversy. Uh Mostly not as much from patients as from medical professionals who were like, "I don't want to go in there and tell these people that, yeah, you have HPE type 3 is the Sonic Hedgehog mutation. Yeah, sorry." Like it like clinicians didn't feel comfortable making that, it was like making light of something that was very serious. I don't think people care anymore, but definitely in the 90s this was a thing. Um and Yeah, it's not the best, but at the same time you could say SHH. You could say like the abbreviation. They would say, "Ma'am, you have HPE type 3. It's a mutation in how? I mean Sonic Hedgehog. Like they can't say it. Sonic Sonic Hedgehog." Yeah, I think that became more the general consensus and the idea that you can use the acronym. But I don't know. I mean, we're scientists, right? We're not gonna stop ever. There's there we're gonna we're gonna keep doing this. We're gonna name a protein Pikachurin, even though we didn't even need to. Pikachurin, the protein named after Pikachu, its gene is egg flam. Why did we? Egg flam is funny. Egg flam is hilarious. Why did we change it to Pikachurin? We didn't need to. Um, and we're just not gonna stop. Even if we name our protein literally Pokemon and the Pokemon company comes after us, that really happened. There was a group of scientists who discovered a protein and they called it Pokemon, and the Pokemon company fucking went after them, like tried to sue them. And so now it's called something lame like ZBTB Pooby Pooby or something. But like, you can't take this away from us. We have Spock 1, we have Frizzled, we have Hunchback. We got lots. We got Smurf. We we have a protein called we have a protein called MAPK, and it gets acted on by I should say the whole thing. MAP kinase, and it gets acted on by MAP kinase kinase, and then that gets acted on by MAP kinase kinase kinase. And there might even be a 4 kinase one, I don't know. The point is, we like having weird names for our genes. It's better. It's more fun. We'll never stop. Oh my god. So yeah, I mean, you guys did it. I know this was a lot rougher than dopamine and vitamin D. Uh it's a very it's always going to be kind of a novel field, even though we first technically discovered Sonic Hedgehog in 1980. We are still putting together like all these pathways and all of these functions that it could possibly have. Um and I mean, the best way to discover the function of like a gene or a protein is usually to make it fucked up. So probably scientists give a lot of mice holoprosencephaly, like intentionally. But yeah, this was very dense, and development doesn't always seem like it's gonna be like really crazy, but it is a very complicated it's very complicated, and the last thing I wanna show is the website where I got those slices from. If you are at all like interested in viewing like all the many stages of human development and like how this site was just amazing. I want to shout him out. It has to do, okay, here we go, with Carnegie stages, which is um the name that we give specific, it's like a general way to categorize the different stages of embryonic development. So let me just show you guys. So it shows you over here like where in the embryo the slice is, which is cool. And obviously this is the later one. The final Carnegie stage 23 is the last part of embryonic development. But like the earlier stages are really hard to visualize, so I don't even know how they got these images, because these are clearly like real human embryo like cells right here. And then I have no idea how they got these. I guess ultrasound, like you could do like a 3D ultrasound, but this shit is so fascinating to me. Page 8. So this is like the groove right here where gastrulation starts. These are the starts, I think, of the like placenta veins or whatever. I mean, this shit is crazy. I love it. But yeah. We did it. Thanks, guys. I appreciate it. This was yes, serial sectioning. Yes, exactly. It's like what they do when you get an MRI and you can like you flip through like here, 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 here. And then let's do sagittal. Here, 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 here. Yeah. Yeah, this was awesome as always. If anyone has any questions about anything, or if you want me to go back to any of the slides, I am willing to do that. I'll be here for a little bit.